We'll wait just a minute or two and see if anyone else pops in a little bit late. Got my big coat, a little oversized. Saw a tail wagging in the background. It's really exciting. There's a dog doing this too. Can you hear me all right? Can you hear me? Oh, great. This isn't your first theme. Great, let's get started. So, welcome to Microscopy 101, learning how to use microscopes at home. It's a STEAM event that's brought to you by JUMP Simulation, University of Illinois College of Medicine in Peoria, and OSF St. Francis. What's really cool about this is that you get to keep the things that are in the kit forever, which is really awesome. So you can explore and learn a lot of really cool and fun skills over the next 3 hours roughly. And then all the add-on packs, you can just keep going and going and going. You can take anything from around your house, pop it under a microscope. If it's too big, you just slice it down. All those things that maybe you were curious about looking at, but never tried it, like maybe mold growing on an orange. Maybe it's just me, I'm just curious about things like that, but you can take some of that mold and check it out. Dust under your bed. You might not want to know what's in that, but it's always something that's fun to look at. Water right out of your faucet. You don't want to look at that. It's not so great. But all the things that you couldn't see before, now you're going to be able to see them. So we're really excited to be able to do this with you. I'm Shannon Egley. At JUMP, I'm the anatomical coordinator at the University of Illinois. I'm an a clinical associate of anatomy. So I teach anatomy to med students over there. And then I work with them here at JUMP once they become doctors to get better at their surgeries. And let's go ahead and get the book open. You can see over on the side of the screen. You can see the screen with the book, right? Great. So I'll be able to flip through that. You can just go to first page. So think like a scientist. I always love to start stems like this because a lot of people, especially me, don't think they're a scientist or didn't think they were a scientist at one point in time. So what's interesting is that we were all scientists as soon as we were born. We were creating these new thoughts and ideas and wondering what was going on with the world. And we had all this new equipment in our body, our brain, our ears, our fingers, our toes, all these things that we hadn't been able to use. And as soon as you're born, you want to use all of those and explore the world. We'll talk a little bit about babies and their eyesight and how they learn about the world too. But down in the bottom corner, I put some QR codes on here. So I'm not sure if you can see the thing, but you can click on the QR codes later. And this is a little video about actually babies and how they think scientifically right from the get-go. So the way that they look at different situations and they evaluate, they actually do their own kind of math and everything from the time they're born. And I'm kind of excited about talking about this right now because during this class or course, you're kind of like a baby again because you're going to see things you've never seen before. It's like a whole new world you haven't been able to be in. So with this course, think like a baby. Be curious. Look around the world, that's already in front of you, but you just can't see with your own eyes. Definitely think like a scientist. 6 steps of the scientific method. So we don't really think much about doing these all the time, but we do anyway. You observe things, you look at things that are in your environment and you wonder why it works the way it does. So you may ask yourself a question out loud, or you might write down in your journal or notebook book or something about, I noticed this happens, I wonder if, and you go from there. So hopefully you see a lot of things now after today that you wonder, I wonder if. Like things maybe you didn't look so closely at before, or you couldn't look very closely at because you didn't have the right tools. So maybe you were looking at fingers later and you wonder what's another part of your body, like your arm, what's it look like? So can you move the microscope to see something there, or maybe pull a hair out and put it under the microscope and check it out. But things that you weren't able to see before, check out all of your fingernail really close. It's actually pretty gross, but kind of cool at the same time. I think the microscopic world is gross interesting. It's gross because it's full of these weird things that you've never seen before, like skin up close. I mean, it doesn't look real that pretty. It takes us getting back at a distance to really appreciate it. But ask yourself questions about what does it look like? What does a pore look like? Or what does a hair look like? What does the edge of my fingernail look like after I've just clipped it with my nail trimmers? Ask yourself a lot of questions, things that you hadn't thought about. I love doing something a little bit different every day, whether it's brushing my teeth with my opposite hand or trying to read something with a mirror or something that's interesting or cool or fun, just because I like messing around and doing things new. So you observe things, you question it, you wonder if, what if, make your educated guess. So your hypothesis is you saying, I think this is the way it is because. So you can say, well, when I get really nervous, I sweat a lot on the palm of my hand, but I don't really feel the back of my hand getting all that sweaty. So run around the room or something, get a little bit sweaty, and then look, do you see any sweat balled up on the tip of your fingers? Do you see it on the back of your fingers? Your hypothesis is, well, since I only feel it on the front of my hand, the anterior side, then I must only have sweat glands on this side. Test that. Get all sweaty, look at the back side of your fingers and see if there's sweat actually balling up or coming out of the pores there. That's the experiment. And then of course, collect your data. If you see sweat on both sides of the hand, you're wrong, but maybe it's just part of the finger. Go on to a different part of the finger. Always come up with a new idea, or recreate your questions and move on to the next thing. All right, there are 3 tools that are really important to have for the course. The first one is your brain. So the right tool is your brain, #1 all the time, because if you don't think the right questions, it doesn't matter what kind of tools or cool stuff you have, you're never going to be able to use it properly or to get the most out of it. So be inquisitive, ask a lot of questions. If you ever need to reach out to me because you have a question that you can't figure out, then I'm glad to help. If you can't get a hold of me, Google seems to know everything. So it's always something that's nice that even if I have questions, I can say, Google, what if this, or what if that, or how many of this happens at this time? So Google knows a lot. And hopefully, since you have a microscope camera or a mount that you can hook your phone up to, you can take pictures. What's really cool about Google is that you can take those pictures and search the web for whatever you just took a picture of, and it'll find things that are similar. So if you see something swimming around in your water from your sink and you snap a picture of it, zoom in on it, isolate that one thing, and then put it into Google Images and see what Google pulls up. Google may actually pull up that little piece of bacteria or protista or whatever it is that you're looking at. So you don't have to have all the answers. You just have to have the right questions, and the rest of the answers will come to you if you know your sources. Right, next, microscope safety. And this is really important. This is not one of the tools, but before we open up your tools, you really need to know how to use them. So the first thing with tools, sorry, I'm going to flip my page. Oh, I can see it up here. I still sometimes have issues with digital things. There we go. So always work within shouting distance of an adult. The microscope is actually a pretty safe tool. It's a precise tool. It's an incredible tool, but you can get hurt with it. Not so much maybe if you drop it and you stub your toe, maybe. But the glass slides are super thin. If you break one, even from a short distance, they could break and they leave a really, really sharp, razor sharp edge. So you want to make sure that if you have, you're working with slides, you want to make sure that your parents or responsible adults close by in case you do cut yourself and you're bleeding pretty bad. Second, keep a trash can, a sharps cup, and paper towels nearby. So the trash can, I say that because of things like paper towels or lens wipes or little specimens that you're throwing away. They can all go in the trash, but not the glass slides. If you do break a slide, you need to have a cup. And I like keeping a little plastic cup, or I actually have this fish bowl too. But with the plastic cup, if I break a slide, I can just put it in the cup, sit to the side. And ideally what you want to do is have a jar somewhere that has a lid on it. So later I can take this to the jar and dump it in, and I have this collecting jar. When it's full of glass slides, I can recycle the glass jar and the glass slides all at one time. So, or you can, maybe a baby food jar is probably too small, but something like a small pickle jar or whatever that you can put in your microscope. You want to make sure that whatever you break that's sharp goes into a safe place. Paper towels nearby, because if you spill, you want to be able to clean it up fast. The microscope, if you spill something on it, there are a lot of little cracks that the fluid can go into, so you want to be able to swab it up as fast as you possibly can, which is leading to #3. Never eat or drink near your microscope. I'm going to be talking for a long time, so I have to have something to drink, but I keep it far away from the microscope. So if I do knock that bottle over or somebody knocks it over, it doesn't go on my equipment. The other thing is that if you're putting something that you think is a germ on the microscope slides, you don't want to be drinking or eating food while you're working with that, just in case it's E. coli maybe, which E. coli, a couple of them really don't hurt you, but if you get it into you and it starts replicating, then it could be really bad. And actually in the bacteria section, we'll talk about E. coli too. Next, handle chemicals and specimens with care. If using potentially dangerous or infective agents, wear gloves, glasses, and mask. You did not get gloves, glasses, or mask with this kit because you're not working with anything that's dangerous. I'm trying to think of the most dangerous thing, maybe iodine, which we put on our skin to stop an infection. Hopefully you know not to drink the iodine, but if it spills on you, you just wipe it off, go to the bathroom, and wash the rest off your hands. It's not going to hurt you if you get it washed off. But if you do work with something like you have some stinky pond water, you never know what's in that. Just put some gloves and some splash guards on. Next, keep 2 hands on the arm and the base at all times. So I already set my microscope up. But when you lift it, just like this, here's the arm over here on the side. The base is the bottom. I love this microscope. I looked at a lot of microscopes to try and find the right one, but it has the handle. A lot of microscopes you have to grab all the way around the tube, and it's just awkward. So this has a perfect handle, and then the base. The reason they say hold the base all the time is because if the compartment down here, if it pops open and things fall out, or if something's loose and the base actually falls off, that would be a bad thing. So it's always best to carry with 2 hands, arm and the base, and we'll go through all the parts of the microscope actually on the next page. Next, keep the scope covered even when it's not in use. So you have a microscope cover that came with it, use it. If something happens and this tears, get like a 1-gallon Ziploc bag or even a grocery store bag or something and keep over just to keep the dust out, because dust is one of the worst things that can happen to your microscope. You usually don't think as dust as being a dangerous anything, but when it gets on the lenses, when it gets on the slides, when it gets on any of the parts and you start rubbing it, dust is actually abrasive. It's like a little microscopic sandpaper. And remember, you're looking at the microscopic world, so everything is exaggerated. You scratch the lens, any of the lenses, and it's going to be exaggerated. You're going to see that scratch forever. And those are really expensive to replace. So make sure you keep your microscope protected and dust off of it. And we'll talk about how to clean the lenses later. Keep the scope covered when it's not in use. I just said that. Always start with the stage furthest from the lens. Okay. There are 2 schools of thought with this. Some people say this is the stage, this black part. Some people say keep this lens as far away so that when you start looking and you turn the knob, then you're slowly bringing in. Some people say bring it as close as possible so that there's no way that you can actually go through the glass slide and break it. Because if you look, this lens here is pretty long. So if I turn it like that and I started going down, there's a chance I could break the slide. So I like keeping it furthest from the stage so I have some clearance and I slowly go down. But like I said, some other people say start at the closest. My advice is start at the extreme. So you'll get used to it. It's kind of like riding a bike. A microscope is kind of weird and awkward to use at first, like maybe an Etch-a-Sketch, where you think you're turning it one way, it's going the other way, and it's just crazy and you're trying to figure it out. But after a while, I've got a friend that can zip through and draw the entire United States on an Etch-a-Sketch, and it takes him like 5 minutes. It blows my mind. Saw him do that at a get-together one time, and I thought, no way that's possible, but he did it. But it's the same thing with the microscope. Once you get used to it, you'll be able to put something on there and find it right away. It just takes a little practice. Right? So you pick the way that you want to do it and then stick with that. Keep the scope covered. Did that one? Always start with stage first. Did that one? Never take your microscope apart. There are a couple things I'll show you to take off, but other than that, you really don't want to take it apart, because if you take it apart and a lens pops out, you don't want to flip the lens in backwards. You don't want to put it in wrong. You don't want dust getting up inside of there. And if you have to replace any of the lenses, they're pretty expensive. In fact, one lens is usually more than the whole microscope costs. So I was lucky that we found a way to get a really good microscope that's normally really expensive at a really low price for you, so you can keep it forever, whether you're in grade school or high school. And in fact, I would have used this in college, so it's something that will last for a long time if you take care of it. And then the last one, only use lens cloth on the lenses. You have a packet of lens cloth. And I'm unfortunately one of those people that's usually like, rules, schmools. I have glasses and I use Kleenex on them or wipe them on my shirt or whatever, and I know it's bad for it, but remember, this is a really good microscope. Don't scratch the lenses. This packet comes with lens cleaning paper, which is 100% grit-free. Even something like chem wipes, if you've been in a science lab at school, these things are called chem wipes. And I always thought I could use these on microscopes or anything, but these are only 80% grit-free. So there's actually particles in here that are abrasive that can scratch your microscope lenses. So anytime you have to clean it, make sure you use this for the glass components. If you need to just wipe down the handle or the base or any of the other parts or the stage itself, you can use a Lysol wipe. You can use just regular desktop cleaner or anything. It's fine. But when it comes to the lenses, make sure you only use lens paper. If you run out of this, you can get it online anywhere, any science store. You can also go to photography stores, and they usually have lens cleaning paper. Okay, and then at the bottom, if you are interested in other ways to clean and take care of your microscope, that QR code will actually take you to a website from a science school supply company that they walk through your microscope again and show you how to clean every bit and piece on it. Oh, that's on my page. There we go. Cool, best part. So now you get to open up your equipment. Let me flip over. I'm sure you've already taken it apart, looked around in it, and found out what you've got, which is exciting. Well, why can't I flip? So good. There we go. Boom. I love this view. Okay, so you should have this black manual too. If you don't have the black one, check your mailbox. It's probably sitting in your mailbox because I had to drop a few of them off yesterday. This is the newest manual. So the one that's just folded up is older. This one's great because all of your add-on kits, you can print them and just slide them into the back as you go, and you'll always have all of your stuff nice and tidy. Another thing I like about this is that some of the pages are like, well, recipes. So if there's a page that talks about how to make a certain kind of slide, you can open this up, pull it out, and put it all the way in the back so you have your own recipe book in the very back. Your kit, in the box, it looks like this. Right. And right on the top of it, you got a little box that's full of random parts. Walk through those. What I did is I actually went to a local hardware store and I found a really inexpensive plastic toolbox. So I put all of my tools on the top, inside, all the things that I like to play with. All the odd things, like I was walking to work the other day and I thought, ooh, I want to look at that plant on the microscope, and I put it in here. So I'll have to soak and rehydrate it. I ran out of time. And then the microscope itself comes in the bottom. If you do get this, this was like $6 or $7. I think regular price is 10. They're pretty cheap. But I put transparency paper on here. Microscope itself. And on the bottom, ooh, I wish that would zoom in a little better. Great. I put some packing bubbles on here. So I have a cushion on each of the sides. And then if I drop this, it's not so bad on the microscope. And even when it's in here, I keep it covered. I'm actually going to put this back. No, I'm not. The rest of our slide set. Great. You have your book. Walk through to make sure you have everything you need. If you don't have something on here, if for some reason you don't have it and you can't do one of the exercises with us, it's okay. We're recording this, so you can go back and do it later. But if you don't have it, just get a hold of us so we can get you that replacement part as fast as possible. So things that come with it, of course, number one, microscope. Right. When you put your microscope away, you always want to make sure that the lenses that are here, these are called ocular lenses, or the eyepiece is always out of here because they don't tighten down in here, they just rest in here. So if you turn your microscope, it'll dump out, and if it rolls to the ground, you could crack that lens, and that would be really, really bad. So you have your microscope. You have a lens set. And the lens set has three, they're called Barlow lenses, and we'll explain those later. But there's one long tunnel here, tube, that actually has a lens on the end of it. And I keep them in the baggies all the time unless I'm using it. You have a 10, WF is for wide field, 10 times, so magnifies right away 10 times, and you can actually see the magnification if you just hover over print. And you can see that in the video. Which is cool. Something that's really interesting we'll talk about later is what that actually does when you get it out of focus and take it further and closer away. That's what this lens does, is it lets you adjust. So I guess we'll talk about that later. Put these pop together. So now I have this ready to go in my scope. So I'll just slide it in. Long tube first, and then what we call the ocular eyepiece, the part you look into after that. And then you have one more lens, which is a WF20X, wide field. Wide field just means that it makes what you can see in the microscope more area you can see better. 20X just means 20 times magnified. Those pieces. Next, you have the phone mount. Actually, one of the things I was most excited about, I looked at a lot of microscopes, but I had to make sure I got one that came with a phone mount. So you can get a really nice camera for your microscope that slides right into the eyepiece, the ocular piece here. I have one, and I got an inexpensive one that was like $75 or $80. And this piece that comes with it, when I just took an old cell phone and actually a broken cell phone up, I get the same quality pictures as I get with that camera. And that camera takes special hardware on my computer. I don't need all that special hardware. So camera holder slides right on. And you'll notice that it doesn't fit if you don't have all the pieces in right. So we'll just hang that there for now. And I'll show you how to work it in just a little bit. Prepared slide set. You actually have two sets. This I wouldn't open yet. When you look at it, I usually do this. I can't believe I haven't marked this box. But it's really easy. For one time, all it takes is you're not paying attention. You open that upside down and it dumps everywhere. So I would get your pencil out, which is actually one of the next things we get. Pencil. And hold on to it. See where the clip is at. When it's in the proper position, oops, you'll flip it up. You know, that's the top. I usually write top just in case, so I know where it's at. Or you can put a sticker on the top, or you can do whatever you want on the top of your artistic draw. When you open it up, always keep this foam in here. It protects the slides and helps hold them so they're not too tight. So if they do drop, it helps protect the slides. So don't get rid of the foam. And there's a list of the 50 slides that come with it in here. Sometimes it's not perfect, but you can see the numbers right below the slides. So you kind of get used to guesstimating. I'm always proud of myself when I'm going like number 36, and I grab 36, even though I can't read the numbers below. When you're a science geek, the little things make you giggle. All right, so that's one slide set. The other slide set is a little box like this. And I did write the top on it. When you open this, you push down the bottom half, pops open. And again, if you open it the wrong way, they'll fall all over the place. And I know I replaced it. I broke one of these because I wasn't paying attention. Keep the foam in there. It helps hold them tight without smashing them. You have 5 prepared slides and then 10 plastic slides back here, clear plastic. And then some with all the add-on kits, you'll actually get some glass slides too, that you can pull the plastics out and put your own glass slides in. It's a second. So we have those blank slides. Just mentioned that. Slide covers. It's a little tiny white box like this. I wouldn't open it up right now, but when you do open it, it's kind of hard to peel open. The glass in there is super, super thin. So a slide is thin glass, but a cover slip is extra thin. The cover slip, when you put it on, I'll show you one step at a time, but you'll never put too much pressure on it, and you'll never force it to go into position because it will snap real easy. Pretty fragile. Okay, transparency paper. This was the best. So the transparency paper, you can cut it however you want. I'll show you how you can cut slides out of this so you have a cheap disposable slide, because this sheet was whatever, 10, 20 cents for a piece of transparency paper. And I have potentially probably 50 or more slides that I can get out of this. So this is really handy for that. And you can even make cover slips out of it too. So it's good to hang on to. If you're going to keep something, if you catch something, you want to put it on a permanent mount and keep it forever, I wouldn't use the transparency paper. I would use glass slides, so it makes it more permanent. I'll show you how those work. Right. Ring stickers. They look like this, or whichever one you got, this or that. But the ring stickers, they're actually going to be for holding droplets. We call this a well. If you put this on the slide, it catches water or powders or anything. So it's like a little well to hold it. Right. You have 6 files. I only have 4 on mine right now. I took the other 2 out. But you have brine shrimp. So we'll talk about how to rehydrate those later. Gum media for working with slides. We'll talk about how to use that later. Yeast and sea salt. So the sea salt and the brine shrimp, you'll create a hatchery. You'll let these incubate over a couple of days, and then they'll actually rehydrate and reanimate. The yeast, I gave you a pack of sugar too to help reanimate that, but you already have 2 live specimens you can work with. With it, you have a hatchery. It's like a tiny petri dish. It's good for keeping specimens in. So you can actually hatch your brine shrimp in here. You can catch an ant in it. You can't get it underneath the microscope, which stinks. But if you have a thin layer of like something you want to look into, like maybe a little pond water or something, you want to cover the whole bottom, that will slide underneath and you can observe it. If you want to look at, I think of weird things to look at, but Jello at different depths. You can take a little bit of Jello when you're making Jello next time, put it on the bottom and drop things into it. You'll have 2 more vials. One will be methylene blue and one's iodine. If they're not marked, it's easy to tell because you take it, flip it over, shake it a little bit, and if it's brown, it's iodine. If it looks blue, it's methylene blue. So 6 of those, your hatchery. Lens tissue, I already talked about this. Super important, make sure you know where it's at all the time. That's another reason I like to have a toolbox, is because I can put everything right back and I don't have to worry about anybody getting into it, and I can put a padlock on it if I wanted to. Even though it's plastic, somebody could break into it. Lens tissue tweezers, forceps, pickups, whatever name you have for those. A pipette. It's also called an eyedropper, like this. So when you use it, and we'll talk about it later, you squeeze it a little bit, you suck up whatever you're looking for, and then pour it on. Another good thing to have, and I'm going to make a list here, you should have a sharps container. So like a sharps jar. You should keep paper towel. You should keep a trash can. Another good thing to keep handy, Band-Aids. Rubbing alcohol. So rubbing alcohol will help sanitize. It's just like putting hand sanitizer in your hands. It's basically rubbing alcohol in a gel form. But if you're using this with pond water, you don't want living organisms in there so that when you go on your next specimen, it's contaminated. So you'll just suck some of the rubbing alcohol up in here, swish it around a little bit, squeeze it out and let it dry, and it will kill all the organisms. Then if you have clean water, you can suck that up a couple times and spit it out, and you've got a clean tool again. So that's handy. For using dyes, you can use that, but I would actually recommend toothpicks, which are in here. Was it next? Nope, not yet. Paintbrush. Paintbrush is for helping if you have like powdery specimens, you can brush them away. If you're doing fingerprinting, you brush it away. Next one was a spatula. So the spatula, it's not super sharp, but it can cut things, so it's handy. It's also good for scooping. It's great for smearing too. It's flexible, so you can get whatever specimen it is and kind of just drag it across and spread it. Pick and probe. So this is handy. You can leave the cover on if you want to, or if you need to push something apart, you can take the cover off and you have a sharp tip. It's not so sharp you're going to hurt yourself on it, but the same thing when you're done with it, you'd want to clean it with alcohol. Next, a pencil. You already have that. You're checking things off with it. A marker. You actually have a marker set, just like this. Open it up. You have the marker. You have a ruler. And you have labels. Play with that just a little bit. Next, toothpicks. Probably have 5 or 6 of them. Doesn't matter how many. You can always get a whole box of 500 at the dollar store for $1. But these are great, like I said, for dyes. When you're using them, just the tip of the toothpick can usually spread a dye really easily. Another thing that might be handy to have, be coffee stir sticks. Coffee stir stick. So instead of toothpicks for spreading dyes, you can actually put the pretend this is it. You can put the stir stick down in the dye, put your finger over the end, bring it out, and just put 1 drop on top of your specimen. So it's really handy, and you can throw them away when you're done. If you're doing it with the toothpick, it's the same idea. You just take the tip of the toothpick in and just put a drop on. So coffee stir sticks. What was next on our list? Toothpicks, pencil sharpener. This is handy for sharpening your pencil, but that's not actually the reason I put it in. This doubles as a microtome. A microtome is a slicer for microscopic things. So with a little tiny screwdriver, you can take this screw out, flip the blade over, put the screw back in, and you can take, let's say you're looking at a carrot. You can take the carrot, put it on here, and just scrape across. If you work in the kitchen, it's like a mandolin for slicing food. But it's safe this way. You flip it over and the blade's up. You take that carrot and slice it, or an onion and slice it, or whatever you're trying to slice. When you go into some of the add-on kits, I'll show you how to take like a gnat or a fly, put it in wax, and when it's hardened, you can slice it across here, and it'll take thin slices of the ant. You can look at it under the microscope too. But more than a pencil sharpener, actually a microtome for slicing. You'll need some clear nail polish. Uh-oh, I moved mine out. Miracle Dry, Quick Dry, whatever you want to call it. A lot of the chemicals to use with a microscope are expensive. A lot of times I find things in inexpensive ways, like at the dollar store. If you run out of this, this is a really great sealer. It doesn't last as long as some of the other things to mount. If you're looking to mount a gnat for 5 or 6 years, this will probably not hold it for that long, but it will hold for probably a year or two. I haven't found out exactly how long, but when we use it, it holds up pretty well. So this is handy for preserving specimens, and it works with your dyes too, which is great. Clear polish, clear tape. This is another type of microscope slide. That's why the tape's in there. You're not taping anything down. It's actually, we'll get to the page here. It's actually clear. It's nice because you can stick specimens on it and you can put it right underneath the scope and look. So I'll show you how that works. And there's actually a fun thing you can do because this is polarized tape. So we'll play with it later. Clear tape, microscope cover. We already talked about this. Always use it. 3 AAA batteries. Hopefully you're already found those and tested it out. On the bottom of the microscope, just a battery cover. You see I already put some in. They seem to last pretty well, or pretty long. I don't know how long exactly they'll last. They'll definitely get you through these courses. So lab notebook and guides, you got your notebook, guides. If you want, a good idea is to have an extra notepad that you can keep your notes in, more like a journal. This is great. You have one blank page, always on your right-hand side, but you might want to transfer information over to a notepad or have your own notepad that you can put a table of contents in the beginning of. You can do it with this one too, first page. But just so that you have your own set of notes that are handy, because once you're really good at using the microscope, you probably won't use this book anymore, but you'll always have your journal or notepad. So that's super handy. And you also have a bookmark and a couple other random little tools that I put in that we'll go through as we need them. So, oops, that's your second tool is your lab. First tool is your brain, thinking creatively, innovatively. Second thing is your lab. And the third thing is your lab notebook. So moving on to the next section. So your lab notebook is really important because, like I said, it's where you write down all of your ideas. Don't limit yourself. I do this all the time. Maybe it's a good thing or a bad thing, but I think it's super handy. I'll be working on something and writing my ideas down for that, but then something random comes into my head, just go to the bottom and write it. My notebooks, actually, if you flip them over and backwards, it's all the random ideas. So my organized thoughts are this way. If I have a random, oh, cool idea, you have to flip my notebook over and start from the back and go in. But don't limit yourself to just what you're thinking about now. Make sure that you're keeping an open mind. I used to do this with my college students all the time, is I would tell them, when you're studying for a test, don't think linearly, thinking this is the next thing because it makes sense. Think kind of just in all directions. So if you have, well, you do have a pencil and a paper. What I have them do to help them think about how to study for tests is that you would take a keyword. Like if I'm talking about muscles, I would write muscle down on my paper, and I would actually start branching off and give myself 10, 15, 30 seconds to come up with all the thoughts that go with it. So if I started, I would say, oh, cardiac muscle, cardiac. I would say smooth, skeletal, red muscle fibers, white, Arnold, you know, and I would just keep putting all the connected thoughts. Why Arnold? I think of Arnold Schwarzenegger because once upon a time he was a really muscular guy. But don't limit what you're thinking. Put down whatever comes to your mind. It really helps. When you get a multiple choice question on a test, if you see the key word in it and it's muscle, these are the things that should pop up. And if you look at the possible options and only one of these things is in the options, go with it. Trust your gut. This is the way your brain thinks. Your brain doesn't think step 1, 2, 3, 4, 5 in that order. Your brain thinks random thoughts all over the place. So a notebook's a great place because you can put those random thoughts down. I put some links to some things about Leonardo da Vinci because what's really cool about him is that when he died, he had 7,000 pages of notes and over 7,000 7,000 drawings. They weren't just anatomical. And they weren't always connected. He did the same thing. He wouldn't have just this one page that had muscles on it. He'd have the muscles here, but then he had a random note over on the side. And when the people that were looking at his books, started putting it together, they were finding out that some of these things he wrote to the side were actually significant, in like the Mona Lisa or the horse that he had built, or even the way that he made his own paint when he was painting the Last Supper, which was not a very successful thing, but that's another thing. Write down your mistakes too. Write down the things that don't work just as well as the things that do work. But notebooks, super important. Keep your mind broad. Write down all the things you come up with. Try this. In fact, we'll take 10 seconds, not very much time at all, but I'll give you one word to write down here. Are you ready? The word is apple, A-P-P-L-E, go. All the connections you have. Probably longer than 10 seconds. What did I get? 1, 2, 3, 4, 5, 6, 7. I write slowly. But some of the things, did you put red? Did you put yellow? Did you put green? Did you put computers? Why computer? How about phone? Steve, like Steve Jobs, one of the guys that invented. I put IBM because I think of the competitors back in the day. You might have put Macintosh, Granny Smith. You could have put all these things, but that's the way your brain likes to think. It likes to think all over the place, not just in a straight line. So try that when you're studying for a test the next time. Find the keywords for the test, write them down, and all the connections you have. What you'll find are a lot of those connections sync up with other important ideas. But when you get to the test and you're stumped on that question, flip the page over, write the keyword, and just come up with everything you can. Look back at the options, and if one matches, go with it. Trust me, trust you. Your brain's usually right. You ever done that? You ever second guess yourself because you weren't sure if you did the right thing, but your brain right away told you the right answer, and then you changed it, and then you got it wrong? Ugh. Your brain should be kicking your butt. Okay, next page. So next thing we're going to do is actually get started with playing with cultures. So culturing means a lot of things. It can be reanimating something, bringing it back to life. Like a lot of the things that you can look at in a microscope, they'll protect your cells. If it gets dry out and they're used to a wet environment, they'll build a little egg around themselves until it's wet again, and they'll pop back out. So you can find lots of things in just a teaspoon of soil or a little piece of moss, kind of like that, or scraping off of a rock or a piece of tree bark or whatever. There are thousands and thousands of microorganisms living on these things, and just that little teaspoon of soil that you can reanimate. So you put them in water, you give them a little time to come back out, crack out of their shell, and then you check them out on the microscope. We're going to see if we can reanimate something in just like an hour or two before we're done here today. So we need time to do it. What you'll need is your little petri dish, your hatchery. Inside the envelope that says 101 on the side, there it is. This is 101 on the side. There's a paper towel in there. Hopefully you haven't yanked it out yet. Inside the paper towel, you have a little sample of moss. Your first specimen. There's the focus. Okay, so we're going to try and reanimate any of the living organisms that are on it. What we'll do, oops, that's a really dirty one. That one's less dirty. I was playing with them earlier this week. So take your dish, you need just a little bit of water. So if you need to go get water, I'll just babble a little bit, but I'm going to take some water and put it in the bottom of the dish. I'm not going to make it super full. And then I'm going to look at this specimen just with my naked eye. And I'm just going to look around while you're either getting water or you're looking too. And I'm going to find a good leaf. So I kind of giggle to myself when I say leaf because there are no leaves. It's one of the traits of a moss is it's a plant that doesn't have leaves, but a lot of people refer to these little branches as leaves. So I found one that has some pretty decent leaves on it. Put that in. I'm going to flip it over. I want to get it wet on both sides. So I'm just doing some flipping. Tucked it in. I sunk it. So what I'm going to do, I'm going to take another one. This one has a pretty decent root. So I already looked at the green leafy part. Now I want to check out the root, which is also funny because a moss doesn't have a root either. It's another trait of a moss that makes it different than other plants is it doesn't have a leaf, doesn't have a stem, and it doesn't have a root. I actually call these rhizoids. We'll talk about those when we talk about plants. So I want to do the same thing with this. Flipping it around, getting it wet again, not stuck. You really don't want to leave part of it hanging out, especially this part, because there are usually a lot of microorganisms that live right at the tips where the nutrients are coming in. So where will be the tip of the root. And I'm going to let these two touch. So if I wanted to do an experiment and find out what kind of microorganisms hung out around the leaves and the other one, what kind of microorganisms hung out around the roots, I definitely wouldn't let these touch. In fact, I wouldn't let these two in the same dish together. But I'm overlapping them so that both of them have areas sticking up out of the water. Do you usually find moss underwater? No. Moss is usually on the side of a tree where it's damp and moist, but not drenched, like underwater, or you find it on a rock. If you see something green that looks like moss underwater, it's actually algae. It's not moss. But whatever's living on the moss is just like you. It needs air. If you push everything under the water, you're going to drown it. So we want to make sure that there's some air, there's some place for them to crawl up or wiggle their way up into air. Right. That's the first one. And you can do that with lots of things. If you are going to the beach or you're going to the lake or whatever, I grabbed a bottle of pond water on my way back. It motivated me to drink my whole bottle of water and then get the pond water. And then I tested that too. But you don't have to let it soak. It's already soaked. You can put some drops in here, let it settle a little bit, suck off the bottom. So take your eyedropper and pull off the bottom because a lot of those organisms will drop to the bottom. And then check it out. And we'll do that later. Right. One thing I didn't do, you should do, is label what I'm looking at. So I'm looking at moss. And for you, if you're not using this page in your notebook, you could just write a notebook and put it on there. But I'm looking at moss. Today's 10th, 4/10. And I put it in at 8:50. So that way, I like to keep track. I'll check this in a couple hours and see what's growing on there. If there's nothing growing on it, I'll check it in a couple more hours. Maybe I'll wait until tomorrow. Maybe I'll wait 2 days. But then I can, in my notebook, say how long it took for me to reanimate whatever's growing in there. But what's really nice is we'll be able to see those leaves rehydrated. Okay, so I put my moss here. It's growing. A lot of places you can get samples. Like I said, you can get pond water out of the pond. You can get, if it's been raining for a couple of days, go to wherever the rain sits, the water sits, like I don't know, a puddle. Maybe somebody left a bucket outside and pull things out. You ever seen the little squiggly things at the top of the buckets of water if you don't dump it out? Mosquito larvae, get one of those. You're doing a little favor by killing it. Pull it out and then put it on a microscope so I can check it out. A lot of places to look. Your fridge. So in the refrigerator, maybe take some yogurt, spread it on a slide and add a little drop of water or some dye to it and stain and see what's growing on there. If mom or dad or you didn't put something away and it started growing mold, check it out. Check out the parents first to make sure that they're okay with it. But like oranges, mold that grows in oranges, it's pretty safe, relatively safe. You don't want to eat it, but you can scrape some of that off and check it out under the scope. And we'll talk about how to do all of that too. So here are just some examples: a scoop of soil, how to rehydrate it, how to rehydrate yeast that you have a little packet of. Right. Back on to track. So the invisible world, this is all about critical thinking. Think out of the box. I know it's kind of cliche to say that anymore, but don't think the way that everybody's thought before. Think differently. Disease at one point in human lifetime or our history, people thought you caught diseases because you smelled stinky air. So it was called miasma. If the air got to you, you were sick and you could potentially die. Well, later we figured out that it wasn't the air, it was the bacteria that was causing the stink that you could get exposed to. So don't limit yourself. Now that you're seeing all these microscopes or microbes under your microscope, you're going to be able to, think more deeply about what's on things. Down the bottom, I put a link to a QR code link to an experiment where you can take some cheese, wash your hands really well, put your hands in the cheese and let mold or whatever's on your hands grow on it. Take some of that, put it under the microscope and check it out. Be curious about what's growing on you. If you have stinky feet, put that piece of cheese on your foot. Hold it there for 30 seconds, pull it off, put it in a Ziploc bag, and then see what grows on it. You might be surprised and really grossed out. But think creatively. Don't be limited by what you think is impossible or there's no way it could be. Test it. Be curious. Try lots of things. One of my favorite doctors in history was John Snow, and he was the first epidemiologist. Epidemics are when disease spreads across a country really fast. We're living in a pandemic where it's spreading across the world, all of the continents. But He lived in London, and cholera was killing a lot of people in London, making a lot of people really sick and killing a lot of them at the same time. And everybody thought, well, it was because of stinkiness. It was people in this part of town. It was really stinky in that part of town, so it must be the stinkiness that caused the disease. And he really didn't believe it. He believed in science. And so he took the map of London and started marking off where all the deaths were at. Looked at places where a lot of people were dying, and he tried to figure out what is around this that's not just the smell. Where do people go to get their groceries? Where do people go to use the restroom? Where do people bathe? Where do people go to socialize? Where do people get their water? Because remember, back in the day, they didn't have a faucet. They had to go to a central pump for a few minutes until finally water came out, and they'd fill as many buckets as they could and walk home. And what he found is that it didn't matter where they got their food or where they bathed or where they socialized. The one common thing was a water pump that was in the middle of a lot of these deaths. So he went to the church, who the church and scientists didn't get along so well back then, and said, I really think it's this pump and the water, something about the water is making these people sick. So the church said, fine, we'll take the pump handle off, and no one can get water out of that well. We'll have to go to a different well. And as soon as they took the pump handle off, people started getting better. A lot less sicknesses, and people are getting better, because you know what the cure for cholera is? So cholera makes you vomit a lot, gives you a fever, gives you diarrhea, because you're drinking dirty water. And the cure is clean water. That's it. So pretty easy. When people were drinking clean water, they were getting better. So the invisible world, nobody knew that. John Snow didn't have a microscope at the time. The clergy at the church didn't have one either. They just thought about it, thought what's realistic, what's possible. They came up with these ideas, and there's a QR code that you can go and read about it. There's actually a game there that you can see how he did it. But think differently. Be a baby again. You're seeing a world that you've never seen before. Use your eyes and your tools to really explore it. So, and here are some of the germs. I put the picture in there for you. But microscopes are fundamental to every science. So it doesn't matter what you're interested in. If you're interested in medicine, or you're interested in forensics, or you're interested in doing material sciences, like learning about clothing and how clothing works on people. Textile scientists make a lot of money, and a lot of people don't even know they exist, but they look at things under the microscope, or they look at chemicals in clothing and figure out what's the best way to make clothing. So you might be surprised with all the different places you can use a microscope. I put another QR code to a website down in the bottom corner, and I like playing with this website, so I'm going to pull it up. And there's the scale of the universe. Just click Launch Tool. And hopefully stays in my language. Oh, I had it up earlier. I'll just go back to this. Oh, that figures. Today's the day. It doesn't work. I'm missing a start button. Oh no. Well, the good thing is that I put a QR code on there for you to explore. Oh, there it goes. It's early in the morning. It's slow. Great. So you can use a scroll bar or the little wheel. And you can look at all these other things compared to the size of you. So you can see a basketball. If we zoom out on basketball, it tells you the size. We can go past it. Holy moly, hailstone. Wow, 20 centimeters. That's almost a foot. So 25.4 centimeters is 1 foot. It's a big piece of ice that can fly out of the sky. Hummingbird compared to a shrew. Let's see a worm. This worm. Oh, of course I can't see it. Now we're getting really small. Coffee bean, 1 centimeter. An ant, sleet. Keep going. Whoops, I'm gonna pass some things. Okay, cool. Grain of salt. It'd be fun to look at under a microscope. What's this? Sand. Here we go. Human hair. And can I scroll over? I want to see the human hair compared to that dust mite. This mite is larger, of course, because it walks on a hair. Climbs on it. Here's a human egg. Skin, one skin cell off your hand. We'll be checking those out later. The nucleus, it's the brain of the cell. There we go, a red blood cell. So I like looking at these things because they tell me sizes, and if I see something like this under a microscope, it's like a ruler. So it's hard to get a ruler under a microscope and use it when you're looking at something that's micrometers, which is millionths of a meter. But a red blood cell is about 7 micrometers, and guess what? When you're looking at anything in the body, there's going to be blood. So if you're looking at a slide that has a piece of a kidney, and you're wondering how big a blood vessel is in the kidney, count the number of red blood cells across it, and you'll figure out how big that blood vessel was. So think creatively. Here's your red blood cell in your body. There's an E. coli, a lot smaller. Chromosomes carry your genetic code. Viruses, there's HIV. Holy moly. Go back out. So there's E. coli and HIV. One time I saw a picture of a microscope picture of a flea, and then they had another microscope that took a picture of a mite that was on the back of the flea, and then they took a picture of a bacteria that was on the back of the mite, and then they took a picture of a virus that was on the back of the bacteria. And that's crazy. We don't think about all these things you can't see, but are always there. For instance, did you know that you have mites actually living on your eyebrow? 9 out of 10 people do. So if you think you're extra clean, you're trying to say that 90% of the people around you are not clean. But if you take a piece of tape and put it above your eyebrow for the night, and the next morning pull it off and look at it under the microscope, you might actually find one. Because they come out at night, they hang out at the surface, they get stuck to the sticky tape, and you pop them under the microscope, they'll actually be wiggling trying to get free. Gross, but cool. Okay, complete history of microscopes. We can get away from this now. If I don't shut this, we're gonna have that eerie Space Odyssey music going all day. Perfect. Oop. Okay, back to the manual. Oh, and I'm way off on my pages. So for real, back to the manual. There it is. So complete history of microscopes. Well, not really, but the cool history of microscopes. So of course, magnifying glass has been around for as far as recorded history. Really shows proof that we've used some kind of like polished glass or quartz. Quartz is basically glass, but when we look at sand later, those are little specks of quartz. People were polishing, so sanding or grinding on it to make little tiny magnifiers that they could look at whatever close up. And I always think, would they ever rest it far enough that they would get the sun to focus on an ant, even like caveman times or whoever? But what we found out is if you keep looking down here, we started polishing these magnifying glasses better and better, seeing things more clearly, but there was a limit. You can only polish it so well and see so much. So actually in 1595, father and son took 2 magnifying glasses, like, hey, if one's good, 2's got to be even better, put them over each other and play with them until they found the right distance, put a tube together, and that was the first real microscope. Compound microscope. Compound meaning it had 2 lenses in it. They weren't really good at it. Their polishing wasn't the best, so they could see things, but they really couldn't see a lot in detail. In 1665, Robert Hooke came along, and he actually kind of cleaned it up a little bit. You can see his microscope here in the picture. It was kind of like a big needle, but it focused. There was a lens down here, one at the top, and actually there's one in the middle because you have 3. But it magnified to look at whatever was down here. You have to have something bright. So he took another lens, put it over here so that the sun would come in, or a candle. You could pivot this thing around so the candle light would magnify and cast the light down. So you had light shining down and your vision was down there, everything was illuminated. And he took a piece of cork, so from a tree, sliced it really thin and slid it under there, and he saw little tiny blocks. And he said, the way these are arranged, they look like monastery cells. So like where monks live, or I think of like prisoners, monk, prisoner, whatever. But these cells are all in order and organized. That's what he saw. So he called these little cells. And since then, we've used the term cells because of him. Not long after, and totally unrelated, Anton van Leeuwenhoek, actually, he was a seamstress. He sold curtains for a living, and he liked looking at things close up, looking at fabrics actually really close. And he was really good at polishing lenses and found the best way to polish lenses. So he took a little teeny tiny lens, polished it really well, put it in this little contraption, and then put a drop of water and looked at it, tilting it to see light coming through, and he could see something moving inside of it. And he was one of the first people to actually see germs moving, so microorganisms. He called them animalcules because they look like little tiny animals moving around in there. It makes me giggle when they show a picture of him doing it. He's holding up, looking at the sun, and I'm thinking that's not a smart idea. But you can build a microscope just like this if you take in that packet envelope, you have a note card and a pencil. Just take your note card, your pencil, and poke a hole through like this. I would say go until it's about the size of the color, like this. I have the paper all the way up to where my pencil turns orange. It's not yellow, it's not orange, it's somewhere in between. So kind of orangey. Poke it through. I'm going to flip this over for a sec. Take a piece of tape. I'm really kicking myself for not giving you scissors, so hopefully you have scissors, but you do have a probe. That's what I usually use. I can't get the scissors. I'm trying to only use what you have available too. Do everything I do. Right. Taking a little piece of tape off. Grabbing the probe. Remember, I had a sharp end or toothpick. I don't even have to take the cover off. And poking the tape with a toothpick. Ta-da. I'm putting, so I poke the pencil in this way. I have a rough edge over here, smooth edge on this side. I'm putting this on the smooth side. Just like that. A little bit of fingerprint on there, but that's okay. And I'm leaving the rough edge pointing up because that will actually help me hold my water in there. You don't have to do this, but you can take a ring. I like doing it because it gives me a loop. But if you push the pencil in, like I said, the ring should go right around that pencil hole. I'm gonna make sure the pencil hole's up. So this is missing his light source, and it's also missing his glass lens. We're actually going to create a water lens here. You take your eyedropper, a little bit of water. You can't see what I'm doing on the table, so let me flip this. Blurry. Still blurry. Didn't help. Great. Well, I'm going to take my water bottle cap. I'm just going to put a little bit of water in it because I don't need much water. Which, by the way, would be a great extra petri dish if you wanted to do a sample with that. Take my eyedropper. I squeezed, letting some air out, put it in, suck the water in, and I'm going to put just a drop or two. I don't want the water spilling. Oop. Oh man, that's perfect. So I only needed one drop and it filled it. And I wish this would be clear because you see my fingers underneath. But that water droplet's nice and round, and that's what you want. You don't want it to be flat, you want it to be round. Now, if I want to read some print, I can just, oh, I spilled my moss. Bring it down and over, and I can read with it. You can do the same thing if you had something that was lit up. So if I went to the dollar store and I bought a little flashlight, they have flashlights that sit flat. You don't have this part, but it's something fun to do. Just happen to have one. Now I have a light source. I could take my Microscope slide. Center it. Look over. It's a little bit too bright, so now I have to move both together. That's better. I don't know if you can see it, but I can actually see an ant on there, which is kind of cool. So I'm looking at an angle, and then every time I tilt my card, it gives me a different perspective, but I can see the ant. I don't know if you can even see the ant without this light. Swoop. A little guy. But I can see it looking through here. Not a huge magnification because water is not the best magnifier, but it's okay. But that's the concept. He took a glass lens instead of a water droplet and amplified the light. It's coming through, which is fun. And then down on the bottom is a little story about him doing that. My focus keeps going out. There it goes. Great. Oop. Nope. That's okay. I'll go back to the book. It's too close. That works. Okay, next section. Other types of scopes. We're not going to talk tons about this, but that cork, we're going to look at cork later. But it had little tiny square cells. If you took a ball of those cells and looked at them under an electron microscope, which doesn't use light, it actually uses well, electrons, part of an atom, and sends it through. You can actually see that's what it looks like, one individual cell. This is a scanning electron microscope, so this one goes straight through. Anything you do a scanning or an electron microscope with, it's got to be dead. The electrons go straight through it. This is a scanning, so it's looking from overhead, and you're actually seeing that round 3D image, and then they go a little bit closer, and you can see some of the individual cells that are sitting there connected. So it's pretty cool. And then practical uses for microscopes, we're going to go through like all of these and a lot more as we go on here. And that QR code on the bottom is a website that has a lot of really cool microscope pictures. So you can look at scanning electron microscopes or regular microscopes, compound microscopes too. Okay, next page. I have a dirty work area. So how vision works. How fun. Just happen to have an eyeball on me. So vision works the way that the microscope works, except your eye is special because your microscope, all it does is amplify or magnify signals. It doesn't make the thing bigger, it just makes the image bigger. Your eye does exactly the same thing. It's just like a compound microscope. It takes the image light, brings it in, and then projects it to the back of your eye. Your eyeball is special because your microscope doesn't have a receiver, your eyeball does, built in. So if we look, the white part of your eye is called the sclera, and the sclera actually is blocking or bouncing back all other light from getting into your eye. When light comes at you, it's actually, if it's white light, it's all the colors of the rainbow, which we'll talk about later. You can see the prism up there. If you split it, you can split all the colors of the rainbow. So the white light, the white light that hits the white part of your eye, the sclera, actually bounces the light back. So the only light that can get inside your eyeball goes right there through the center, called the what? It's a P word. Pupil. Yep. But before it gets to the pupil, it's actually going to be magnified and manipulated. So if we take the top of the eyeball off, you can see the inside. Get over here to the desktop. My hands aren't fast enough. There we go. Top view. So light's coming in like this. The first structure it goes through is not the sclera, remember, but it's part of the sclera. It's all continuous. There's no break. When we talk about the integumentary system or the skin, there are no breaks. Everything's folded. So the white just goes straight into the clear. There's just no white coloring. It's called the cornea. And the cornea actually bends light, manipulates light. 2/3 of all the light bending and manipulation happens because of your cornea. But what do we give it credit for? Or what do we give credit for bending light to? It's not the cornea. What's the thing in here that magnifies or manipulates light? It's the same as on your microscope. It's a lens. the reason we don't give the cornea credit, even though it does 2/3 of the light bending, is that you can't bend it with your eyeball. Your eyeball can stretch and squeeze, not squeeze, stretch and let go of, relax your lens to change the shape, but it can't do that to the cornea. So what you see through the cornea stays that way, unless you squeeze your eye really hard and smash it and bend, then you can do that. And if you've noticed, I'm not wearing my glasses today, so I do that with my eyes. People that are squinting all the time, that's what they're trying to do. Their lens isn't working properly, so they're trying to bend their cornea to bend the light differently so they can see better. So the first structure is the cornea. Right behind it is a water pocket. Water manipulates light too. Not a lot, but a little bit. So as it goes to this water, we would call the aqueous humor. Humor is referring to the liquid. Aqueous is it's a watery liquid. It comes through, goes to the next structure, which is the pupil. And this is the iris. The iris's whole job is to change how much light comes in. That's it. So on a bright day, it gets smaller. On a dark day, it gets bigger. You already knew that. But also when you're trying to focus on things, it will open up to help more light come in from whatever that object is that you're focusing on. So it can manipulate the light. Some microscopes actually have a manipulator right here that's called an iris diaphragm because you can change the size of the light coming through. Next is the lens. And the lens, when you're a baby, is pretty round, more like a ball. As we get older, it gets flatter like this. When you're a baby and it's nice and round, you see things up close really, really well. But as it flattens out, you see things further away. So when a baby's first born, what they really see are just things that are close. So if they look at you and they stare at you and you're 5 or 6 feet away, they have no idea that it's you. You're just this thing that's kind of blurry in the background. If you get close to them, then you freak them out. But this is the lens. We can't manipulate our lenses because they're glass. Next, light goes through the lens, gets changed. We have two muscles. We have two sets of muscles that are changing the lens, and another two that are changing the iris, squeezing it in or pulling the lens to make it flatter. This isn't solid in your eyeball. Unless you've taken the dissecting course with this, then it's kind of jelly-like. But this is called the vitreous humor. So this vitreous humor, if it's jelly-like, we could take it out, but it's going to bend the light too. There might be little floaty proteins in there that your vision has to adjust for. But so now we've had the cornea, the pupil, the lens, the vitreous humor, the aqueous humor. Finally, the signal gets all the way to the back of the eye, back here on the retina, where you can detect the signal and see what you're seeing. So your brain has to interpret that foggy, too bright or too dark, kind of messy image that gets projected. If it gets projected straight back, you don't even see anything, because this is the optic nerve. There's no receptor there. Where you're seeing perfect vision isn't straight back in your eye. It's actually over here to the side a little bit, right there, called the fovea. So you're trying to get an image to come in and go slightly over here, oops, wrong way, slightly over here to the fovea, where you get clear pictures on a sunny day. So the fovea is good at detecting colors. So you have special things called cones in your eye that detect colors. They're not really good at night. So the rest of your eyeball is full of things called rods that detect light or dark, just black and white. So at nighttime, if you focus on this color sensing area, you don't see very well. If you kind of tilt your eye out of focus, you'll actually see things better. Give it a try sometime. Another fun thing to do is if you have a hacky sack or a juggling ball or something that has lots of colors on it. Back to this. If you have a friend take the hacky sack, you look straight ahead and you have them bring it from over by your ear and slowly bring it until you can see their hand, have them stop. Try and guess what color it is. It's like impossible. You might guess that it's a light color or a dark color, because what happens, oops, we'll go like this. The light comes in, it goes up here at an angle. So if I'm holding this thing at the side of my head, it goes up here at an angle, and it hits where the light and dark is at, not where the colors are. So I can see if there's something there, but it's just black and white to me, or shades of gray. It's not until I bring it closer to the front that I actually hit the color receiving area. So that's kind of fun. Again, the microscope works a lot the same way, because it has multiple lenses that magnify the image or let you manipulate the image. Some of them have an iris. You can adjust the light coming in. you have a knob on the side, you adjust the brightness, which is on the next couple pages. So vision's kind of fun. If you haven't done this before, I know you've done it. If you haven't gone swimming, you're missing out on life and you need to get out of the house more. But if you go swimming, have you ever noticed that when you're looking down in the swimming pool, your friend's legs are really short? Like their body above the water looks okay, but their legs, super tiny. I'll use this one. So if you have a glass at home, it's better to use a cylinder than a bowl or rhomboid shape. Put the water in. Not all that exciting. I mean, it kind of bends the light, blurs things. But let's say that this is a swimming pool, and then your friend, let's see if I can get into the camera, is in there. Oh, not as exciting. There we go. The way the pencil bends a little bit, right there where the water changes, because light bends in two ways. One, it can be bounced or reflected. So if you see something that's yellow, guess what color this pencil hates? That yellow. White light will come to here and absorb all the colors except yellow. That gets bounced back to your eye, so you see. That's reflection. There's refraction, which means it bounces as it goes through something. So clear things like water or glass or plastics let the light come through, but they bend it at different speeds and at different angles. So the light is refracted as it goes through the water, just like it does when it goes through the vitreous and aqueous humor in your eyeball. Same idea. Lenses are great at refracting. Light goes through them. The glass ones are super powerful at bending. You can manipulate glass really well to bend the light right where you want it to. So your eye reflects. Sorry, your light refracts. Mirrors reflect. Things reflect light back at you. But if it passes through, it's refracting. So you can build another water lens that works a little bit better because it's bigger. In your little envelope, you should have two little tiny Ziploc baggies. Hopefully you do. Mine had it earlier. I dropped them out. I always keep a backup or 300. You can take your Ziploc bag. Looks like it's intact. Looks like it's not broken. Oh, good. And plastic bends light. Plastic's a terrible light bender. That's why it's hard to get really clear plastic. You can take your water and step away from your microscope and everything electronic. And fill your bag. So I have a full Ziploc bag. Here's where it gets a little bit messy. I'm going to do this over a bowl. And I closed it. Okay. Now you have a lens, a water lens. If you look at the tabletop again, hopefully you can see clear enough. Uh-oh, I may have had a leak in that. I look at, oh, I can't see. So I bring it down and I look. Of course, my image is fuzzy. Here we go. Surprise camera. But you can see it magnifying. If I make it more round, it magnifies a lot better. If I flatten it out, not as much. Same way with your lens. The more round, the more magnification you get. The flatter, the less magnification you get. So you can always shoot yourself like a baby. So a fat round lens like this will probably see a couple feet in front of you. If I walked around today, there's this thing on my, like this, oh, I am up there. So maybe if you make some impromptu glasses, you have two of these baggies. Woo! Oh, no. I can see Megan sitting over there. I can see things, but they're pretty blurry. Oh yeah, I can see way better now. I don't need my glasses. I'm doing just fine. But maybe if you want to be like a baby, you could make a pair of goggles like this and spend a little amount of time looking around and seeing how a baby sees, making them your own lenses. Piece of advice, don't walk around the house like that and let somebody else know what you're doing, so that when you go staggering in, you don't, well, you will look like a fool, but you don't fall over and look like a fool. You can see water refracts, glass refracts, plastic refracts, and we actually are going to make a device that shows light bending as it goes through plastic later. All right, we'll just walk through compound microscope and we'll take a break. So just the pieces. So you know what parts are parts. 2 hands. Remember, whoops, let me get over here. Arm in the base. I'm holding it by the base, getting nervous. There we go. So the different parts. The first thing you look through is the ocular lens, the eyepiece. You can take this off, remember? This is 10x on the top, so you can actually magnify with it. You have a 20x over there. Well, I'll show you how those work later. Right. The tube, you have a rotating nose piece. Wrong way. A revolving nose piece, I'm sorry. So that when you turn it, you hear it turn, click in place, your next lens, click. Each of these lenses are called objectives. So you have an ocular lens, revolving nose piece, and objective. Every time that you start, every single time, you're going to make sure that it's on the red lined lens. This is called your scanning lens. So no matter what you look at, the first lens you'll ever look through will always be your scanning. It's the safest, number 1. It's easiest to find things, number 2. So objective, red, that's your starting point. Next is your stage, and that's the black thing that you put your actor on, your specimen, your slide, stage. These little things are stage clips. When you're working with the slide, it's a lot easier. Oh, it didn't break. It's a good thing there's cushion on the table. I just made a comment about that earlier. You can use your thumbs. Okay? So if I'm manipulating a slide up here with my fingers, I can just take my thumbs and they're spring loaded, just push back and lock it down. Very nice. Your microscope actually has add-on pieces that you can get for it. Like the screw hole up here is a little piece of metal that you put on here to make sure that you can't get too high or too low so you don't ever break a slide. You have top light or upper light up here, episcopic light, being superficial. That little hole in the center is your condenser. So your condenser takes the light coming from below and condenses it. It does exactly what it means. This is a diaphragm. You can't adjust your diaphragm really. If you look underneath, don't tilt yours like this if you have the lens in. If you look underneath, as you spin this, the different colors. So all the holes are the same size. The only difference is that you have filters. Some microscopes will have different size holes, so it lets less light in or more light in, depending on what you're looking at. So you want to always keep it on a clear one, unless you're specifically using a filter, which we'll look at later. Right, so the diaphragm with the filters on it. Next are, well, here's the arm. Here are the knobs. You have a big knob called coarse adjustment. If you turn it now, you can see it moves the stage a lot. As I turn it, playing with these, it's just like an Etch-a-Sketch. You just get used to turning it the right way because you just get used to it. The little knob is called the fine adjustment knob. So when you turn it, the stage doesn't move as much. You turn this one whole rotation, and it just doesn't, it takes a long time to get through that distance. When you're using the red objective, the red line is the only time you should ever use the big knob, ever. If you're using a longer objective, it sits really close to your glass. And if you use this big coarse knob, you might send it right through the glass and break your microscope. So coarse only for the starting, only when you first start looking at things. Everything else is fine. I'll show you how these work and we'll practice with them a lot. Right? Next, you have your illuminator. It's a fancy word for a light. And then a rheostat over here that adjusts how much light comes out. So You might think you have to crank up the light full blast every time, but maybe you can't see so much because of the angle, but it's really bright in there. You don't need all of that light. If you don't need it, don't use it. It just makes it worse on you. So back, power switches. So I've noticed that some of them, if you turn on one, it turns the top on. Some of them, if you turn on one, it turns the bottom on. Just get to know yours. You need to take one of the sticky notes that came with your marker and put it on here, those little stickers. You can always write top and bottom or up, down, or something like that. And then of course, the base is the bottom part that holds the batteries. That's the walkthrough through the microscope. When you clean it, what do you clean the lenses with? You only clean them with lens paper. So if I'm cleaning the lens, take a little bit. You can take a whole sheet if you want, but it's unnecessary. I can't get the sheets apart. Don't lick your fingers and do this. Oh, okay, I have a scrapper. Scrap right here. So I just need this little piece on the top. When you clean the lens, you can use a cleaner on it, but the only cleaners you'll want to use are either, well, I use something called Zeiss cleaner that you can get at Walmart, where they sell glasses, or they have the glasses, but that's made for lenses. You can use Windex, but dilute it 50%. So take a little container, put half Windex and half a clean water, not from your faucet, purified water, ideally, denatured water. And then you can put it on there if you want, or you can just do light brushes and then brush the same direction across your lens. I always do the same direction so that if there is something sitting there, I'm pushing it off. I'm not scratching it back and forth like sandpaper. Bring it down, bring it down, bring it down. I'll do the same thing down here. So my objectives, bring it across. This objective sits pretty close to the surface. Same thing, one direction. And the red one you probably won't be able to get to because it sits up inside, but just clean it around it. And then let my light off. That one doesn't matter if you go around. There's one more. Down all the way. I'm looking at it, so I'm using my coarse knob, but I'm looking at what I'm doing. There's a piece of glass. It's the condenser right here. Kind of going back and forth with it. And there's the glass pieces. Clean lens paper each time. And then if you want, I put QR codes to an online microscope, which is kind of cool. You can play with the digital version of it, and you don't even have to have your own. But I'll let you take a break. I'll hit pause here, and I'll see you in 5 minutes. Cloud. Okay, I'm back. Hopefully you're back too. All right, first look. Time to take a peek in the microscope. That's exciting. So what eyed are you? What handed are you? Are you right, left-handed, ambidextrous? So I'm right-handed, but just because you're right-handed doesn't mean you're right-eyed or right-brained or any of those other rights. So one way to do this is if you pick something in front of you, like maybe you're picking my hand, look at it, both eyes. I'm looking at the camera. I can see the camera right in the center of my circle, both eyes. Now close one eye. Did the camera move? Are you still seeing the camera? Close the other eye and open this one. It moved, totally moved. So I'm actually left-eyed. When I close my left eye, my whole vision jumped at that time. It's telling me that my left eye is dominant and my right eye is just helping the image of the left eye. So I'm left-eyed. Doesn't mean you have to do it this way, but which works out really well because I can look in with my left eye and I can write with my right hand. And if you're still comfortable using your right eye, that's fine too. I think I actually switch back and forth quite a bit, but that's a little fun game. I can tell all your friends, ask them if they're left or right-eyed. Okay, when you're using the microscope, you already know the parts, and I'll keep using the names over and over again so you're used to them, but you want to get somewhere that you can sit comfortably. I'm standing at a table that lifts up to me. Make sure that your microscope is sitting level. All feet are on it. Don't sit on the couch with this on your leg or on a couch cushion, or sit on the floor and squat down really low so that you can see and tilt your head back. You want to make sure that you're sitting somewhere or you're standing somewhere that you can get to the microscope comfortably. I don't like to bend my back a lot, so I actually spread my legs apart a little bit when I'm looking at the microscope and bend more with my knees, just like you're lifting up something heavy. Make sure that it's on. So power on the back. Make sure you have the bottom light on, the bottom illuminator, not the top one. Again, with the stage, if you want, you can do all the way up so that you don't accidentally put an objective into it. But what's important right now is that I'm all the way at an extreme. When I put my slide in, there's only one way I can turn this knob. And so I'll turn it slowly, keep turning until I see what I want to see. So by starting at one extreme or the other, I'm always able to move in one direction until I see something. Otherwise, it drives me nuts when I've tried to help students in the past and they go up a little bit and then back down a little bit and then up a little bit and back down. Well, what if what they're looking for is up here and they just keep coming up and down here? So if you start all the way to the extreme and you just go all the way through all the possible perspectives, you'll hit it. Okay. So make sure that I skip anything. My instinct just takes over sometimes when I'm setting the microscope down. All right, we did it. Flat surface, turn the light on. Nose piece to the scanning, which is what color? Red. So it's the red objective. This one. Position your slide at the center of the stage light. I'm actually going to have you do one thing. Since this is maybe the first time you've ever used a microscope, put a slide in. There's something that I think will help. I spilled water. Oopsie. So if I take my slide, and what I would do for you, take the top box. You can use any of the wooden box, but you just take the top box. Remember, this is the top. If I push on this part, pop it open. And one of the first ones that I'll see is cotton. Your first one might be a fly's leg or something, whatever. Close this, put it back. If I remember, it's going to be. If you take your marker, yep, I have a mess. My water spilled. My little cap. There's my stem of cotton. I can see, I know you can see it too. Right there, the green dot, that's my slice. So they cut across the stem real thin, put it on there. I'm gonna look, I'm not gonna write on that with my marker. I'm gonna go all the way to the edge of the slide, right on the very edge, directly below. I'm gonna leave a black mark. Can you see it? I don't know if you can. Really, it's there, and on the microscope, I'll definitely see it there. There's my black mark. So when I'm putting this under the scope, the round condenser hole, that hole right there, I want to get that green dot right above it. So whatever your specimen is, you want to put over that. Lights off. Bottom light. I'm bringing it in, and I'm going to start with the black dot in the center. So I'll bring it in. This is where I use my thumbs. Use my tips of my fingers on my slide. Bring it back. And you can't see this yet, but the black dot is right at the edge of the light. So now I'll squeeze this, bring it back while I'm looking in the microscope. And I'm actually going to find the black dot. I'm not going to worry about the specimen yet. I want to find the black dot. I'm just going to worry about it, focus on that. And then I know once I'm focused and I'm in the right place, I can just slide my whole slide back and I'll see it. One thing I probably should have shared with you, how to set up this. You don't need this for today. We're not going to do anything. But the phone holder is really nice because you can take pictures. Whatever you take a picture of, you can put a Google Images and it'll help find and describe it for you. Most of the things I've looked for, I've been able to find pretty fast. But using this is the part that will mount to the lens. This tightening screw, make sure it's loose but not too loose so that I can still adjust, but it's snug. And I'll put that there. Oops, I have two of them. And then you can see down in the bottom left, or sorry, bottom right-hand corner of the screen, that's my camera. So I want to make sure that my camera is looking down in that. So where it gets tricky, you might want to have someone help you, but right here, when you're squeezing the phone holder, just get that in, twisting and turning all over the place, not good. Wobble, wobble, wobble. I'm trying to let the weight of this hang down because otherwise it can twist back and forth and mess everything up. And then watching, okay, definitely to the left. Go up a little bit. Come over. There's some light. I'm getting really close. Up a little bit higher. I'm looking for the perfect circle. Still too high. Sometimes you get it right away. Sometimes you have to mess with it for a little bit. There it was. I missed it. Oh, too far. Come on. Too high again. Down low. This is just one of those days. Oh, there it is. Nice and bright. And then it's like trying to shape the moon. Full moon. Tighten that screw on the back. Boop. Okay, yeah, I'm looking for that little black dot. Oh, went in really fast, but nailed it. So there's the black mark. That was clear. There's the edge of my slide. You can see the line. I don't have to move very far up with the course. In fact, it's almost focused right now, but I'm just going to mess it up on purpose. So now I'm focused in the glass. Now I'm focused on the edge. You can see how crisp the edge is just by moving that back. If I want to make it nice and clear, I take the fine adjustment and go really slow. You didn't notice this, but I put the black mark under the slide. You don't have to do that. It was probably a better idea to put it on top. I just slipped. So I'm seeing the glass edge, and my marker image is actually on the bottom of all of the glass. But I know where I'm at. The only problem with this is when I go to get my specimen that's sitting on top of the glass, I'm going to be out of focus. I'm going to pull straight back, looking for my specimen. There it is. Now I'm in. Just move around a little bit. I definitely know it's that, so I'll use the fine. I know it's what I'm looking for. There it is. So over on the left-hand side, I'm seeing the bark layer, the outer layer, the epidermis of the tree. And then the cortex is the one that looks more like bubbles. This would be like your skin, cortex. And the deep part, if I can get there, there we go, is the medulla, the medulla. It's pronounced medulla. It's kind of like stained glass. It's pretty cool. So now I can flip there. And if you're looking through, my phone doesn't show it right now because it's plugged into my computer, but if you look at the phone, just fine tune it until you get what you want. Another thing you might want to think about when using the phone is turning off the autofocus so that you can do it on your own and it doesn't try and correct what you have already corrected. You can still have those cells. Okay, so once you find your image and you find what you're looking for, I'm going to go over and look at the edge so you can see the layers. Then that revolving nose piece, these are the objective lenses. I'm going to grab it. I'm on the red right now. The next one I want is the yellow because it's the second longest. So we have low power, higher power. I'm turning it. Make sure it clicks. So what's nice about these microscopes, and you don't get this very often without spending hundreds of dollars on a microscope, is that they're par-central or par-centric, which means that when you flip from one objective to the next, it keeps your center pretty close to the center. Some microscopes, these lenses aren't quite right. So if you get like a cheap kid's microscope, you might have everything perfect when you're on the scanning objective, but you flip it over and suddenly your image is completely off the picture. But all I have to do now is adjust my fine knob, and I'm back. So remember how I wanted to see the 3 layers again? I can see the outer layer, which is all the way on the right. There's a central layer, the cortex, so the epidermis over there, which is the superficial part. It's like the surface of your skin. The cortex is the next layer, and then the bubbles all the way over to the far right would be the medulla. So 3 layers. It wasn't, sometimes it's par focus, which means that it's pretty close to end focus when you flip between the objectives, but this is not. So now I'm going to go from the yellow to the blue, really close. Same thing, it's just a little out of focus. Whoa. My phone's trying to adjust, but I can see the cells now. I need to remember to turn that off. See that kind of hazy weirdness? I'm actually looking in the glass cover slip right now. And now, oops, as I go deeper, now I'm actually looking in the cells. So that's how you focus on something. You get everything set up in focus. First thing was flat surface, turn the light on, rotate the nose piece on the red objective lens. Put your slide in the center of the stage light, which is that condenser coming straight up in the middle. Adjust the coarse focus knob, the big knob, until you see your image. Fine focus, recenter to wherever you want, and then flip the objective, so the next one on the revolving nose piece. Which focus knob should you use? Anything but the red. So now you're on a yellow lens, you have to use the fine. You don't want to break anything, so just stick with the fine. Bring it into focus, and then move to the highest objective, the most powerful one, and then fine tune your focus. If you need to at that point, you can adjust the light. So if you had a diaphragm, you could flip the diaphragm here. Like if I want to make it not so bright, you can dim it down a little bit. Some mood lighting here. Yeah, this one's gonna be better on brighter light. There we go. If you want to play with the filters, you can change those. Red filter. No, red filter is not good for a green subject. I think this one's actually yellow, not green. Green. So anything that wasn't green would stand out as being black if I have the green filter on, and we'll explain that later. but the green light goes right along with the green stain that's on it. So the green really stands out and everything else is not green. Whether it was blue or red would be more of a black color. So it separates everything that you're looking for. There's a QR code if you wanted to look at it about cleaning your lens and prepping things again. Oops. So now let's look at things. You don't have to use slides every time you look under the microscope. You can look at small things that you want to look up a little closer. So I'm going back to the objective lens, the red, pulling my slide out, putting my slide back in my box so I don't accidentally leave it somewhere and break it. When you're putting the slides away, make sure that you get it in both sides of the holder, this side and that side, so they're not crooked, because if they're crooked, it may not rest properly, and when you shut the box, it'll break. Closed. I want to turn this back to the white. Oops, going the wrong way. Blue. That was it. Clear. Now I'm just focusing on, looks like there's some sticky stuff on my stage, my condenser. That would explain why it wasn't all that clear. So I'll take my lens cloth. Cleaner. Tissue paper. I'm going to just squirt a little bit of lens cleaner. I'm not going to make a mess all over the stage. Bring it all the way down. And I have wet material on here, so I am going to kind of circle a little bit instead of just going in one direction, like I'm washing it. Look better. Yep, not seeing sticky stuff. There's nothing to focus on there. Oh, put my slide away. Oh well. We have other things to play with. So drop your stage all the way down, like an Etch-a-Sketch. Remember, just keep turning until it's all the way down. Let's check out your skin. So you look at your skin every day. You see the papilla ridges, epidermal ridges, the folds in your skin. We call those, what do we call those? Fingerprints. So let's see if we can see our fingerprints. Put my finger under there. Oh, there it was. So this is kind of hard because my finger wants to move, even little microscopic movements, you can tell. Hey, what's that? Little cut. A micro cut. My finger out a little bit more. So I was watching the fingertip just now, and now I'm moving to the more proximal, which means closer to the rest of my body. I'm losing, my fingers are too big. I'll use a smaller finger. There's the tip of my pinky. There they are, fingerprints. Don't push down on your stage very hard. Just kind of rest your hand there. Moving along, I'm moving back towards my knuckle. There it is. Gross, dry skin. More skin. You can actually see how the surface of your skin is clear. If I look up at the top, you can see how clear that skin is. Whoops. It's just grazing at the side of my finger. The light's going right through it, really. Maybe flip your finger over, see what's on the back. Too far. Fat finger. That's better. Ooh, gross. There's the edge of my nail. Very nasty. Ooh, something on my nail. Well, that's got to go. See, you can get really clean with a microscope. There's a toothpick. Let's see what your skills are if you have something on your finger. How gigantic that toothpick is. Oh, kidding. Wow. Well, gross. So that's the fold. Here's my fingernail. Oops, if I can find it again. There. Oh, little tiny movements go a long way. Fingernail, rest of my skin, cuticle. Let's see. You can see how clear the fingernail is. It's almost like when we went through the glass, we could focus on different levels. Gross, but my finger looks normal from a distance. Uh-oh, how do I trim my nails? Let's see. Hopefully you're doing the same things. Just play with it. I lost my finger. There it is. Nope, I'm not going to be able to see the fingernail edge. It's the best. The light's just too bright. Let's see if we can turn it down. Nope, still too bright. Oh well, I have a scar up here. Let's see if we can find the scar. Focus. Ooh, there's that dirty spot. Ah, there's part of the scar. Crazy. Fun. If you can find a hair, see if you can check out a hair. I do have hairs, but my hands are too big to get under that part. Yours are probably smaller. But later, come back, explore your fingers. You actually have a fly's leg inside your set. You should pull it out, pop it in, and see what the fly's leg looks like. I can't wait. Let's do it anyway. So, it's in this container. Stem of cotton, pine mature, epidermis, housefly. Sweet. You can do the mark on the edge if you want again. I kind of know where, actually I did already mark that edge. My stage is all the way down. There's the edge of my slide. Shoop, like that. That slide needs cleaned. You can use the same cleaner on your slides. That's the marker right there. It's kind of a purplish color. And you usually put it on the top, so let's count on that being on the top. Slide back. There's the circle, the edge of the cover slip. You can see there are air bubbles underneath it. They're there forever. There it is. Hello, fly. Sweet. The fly's hairier than me. That's nice. So you can see the big vein going down the middle of the leg. It's almost like looking at a crab leg. On the far right, you can see the muscle, the meat inside of it. You can see the shell broken like a crab leg. Focus. Can move along. I'm going to try and look at one of those hairs. So we want to put the hair in the middle. If it's taking you a little time to do this, don't worry about it. I've been doing this a while. A little more light. Just decent focus. I want to go to a higher objective. I'm going to go to the yellow. Click. Out of focus. Turn my fine knob. Cool. Just like a crab leg with those little spiky bumps. Go over. You can see the hair. You see the vein going right down the middle, the blood vessel. So one more. Let's see if I find a good hair. There's one. All right, let's see if I can get it. I'm fine tuning. I can see the surface right there where it's coming out of the leg. I'm going to click one more objective. Spin the revolving nose piece, lock it in. Oops. Fine tune only. There it is. Very cool. Too far. This is probably the hardest part, is moving the slide on the high objective. Awesome. You can actually see the center of the hair. So if you see it, look at the hair, it looks like it's striped going all the way the length. It's actually showing you the 3 layers of the hair. Just like on the tree, on the outside, you have a cuticle on the outside. You have a cortex is the second layer and the medulla is the third. So the epidermis layer, the very superficial part, which means on the surface, is the only thing that changes the name from the tree. But you can see right through. So that's the cool thing about microscopic, is that you can see right through the specimen. The light goes right through it. It blocks and bends some colors of light so that you can see the image. And so if I wanted to, I could take a snapshot. I don't know if my phone will do that one. I'm never going to mess with it. But normally, if it wasn't hooked up to the computer, I could take a snapshot. I don't know if I still can. Might be a little bit dark. I've taken better pictures. And I can go to the website. Where'd that website go? Here. Go to Google. Google Images. I can paste. I think I have to save it as a picture, but then, yeah. Oh. No. So you can save it as a picture, and then upload an image, choose file, and then let Google hunt for it. That's, I should have done a sample one ahead of time. But Google's pretty good. It might not grab necessarily a fly hair, but I'm sure it's going to know that's a hair and then go find it. All right, back on track. So look at the finger, look at the fly. Flip the page. Boom. Analyzing pictures. This is a fun one. So to take the slide out. I'm going to drop the stage all the way down. Pull the slide out. Put the slide back in the box. What's in the box? And I'm actually going to take the page out of the book without ripping it. That's the wrong page. Because we're looking at things we don't need slides for. And analyze some text. I want to see what these pictures look like. So I've got a tiger and a purple butterfly. My orange tiger, I should see, looks like orange, white, and black are the only colors on this tiger. So make sure the stage is down. It is. I'm gonna find myself. I'm actually gonna take the clips and bring them to the side because I don't want them right now. I'm gonna find the edge of that paper. Just went through it. There it is. I can't see it through the microscope, but I see it with my naked eye. I'll bring the stage up slowly. Oh, I know why I'm doing it. Somebody forgot to flip back to scanning. That's why. All the way down. Lights up a little bit. Edge of the paper's on the light. Course adjust, bringing stage with the paper to me. There's the edge of the paper, which is cool on its own. Awesome. Let's see what else I can find. Oh yeah. So this paper is factory cut, nice and straight. There we go. There's a hole, my 3-ring hole. There's the edge of the hole. Follow it around. Pretty sweet. And then my tiger should be right up here. The paper itself is pretty cool. Paper is a lot like plywood. If you go to a hardware store and you see those big sheets of wood, they look like a bunch of chips. Paper is the same way. So I'm gonna go right up. There's my orange tiger. Nice and orange. Actually, I don't see any orange at all on there. I've got to be on the wrong animal. Let's see around. Still none. I see pinks, purples, blues, yellows. Can't be right. Cool patterns. Still no orange. Huh. Maybe if I check it out on a higher objective. That's pretty sweet. Still no orange. That's weird. I'm definitely looking at the tiger. How about butterfly? If I just slide it over, I know I'm in the right plane. You can see the paper again, which is extra cool. You can see the fibers in the paper. So it's not perfectly flat at the surface. So awesome. And I'll look for that butterfly. Bringing it in, bringing it in. Hey, there's my purple butterfly. So I see some purple. It actually looks like most of it's blue and red. I don't know why that is. Well, it's like that because most printers only print 3 colors. It's cyan, which is a blue, magenta, which is a red, and what's the name for yellow? Yellow? Just yellow. Yeah, I don't think there's a special name for yellow, but they're not. So the blue is not the traditional blue. The red is not traditional red. It's definitely not the same, but the yellow is yellow. And what happens is that when we're looking at the butterfly over here, this is what your eye actually does. When I'm close up and I can see details, it looks mostly red and blue. But when something's further away, it seems more purple because those colors just blend together. The light from the reds, the blues, and the yellows actually go to my eye and make me feel like I'm seeing purple. So pointillism was actually a way to paint, or is a way to paint, where you take and make small dots of different colors, and then the viewer standing at a distance, and they don't see the individual dots, they see the blending of the colors in their eyes. And there's a QR code to a really cool illusion that's Marilyn Monroe and Albert Einstein, where if you zoom a focus in on the picture close, it shows one of them, but as you pull it back, it actually shows the other person instead. So one person gets blurry and the other one pops into view. And what they do is they trick the eyes. They take little tiny dots and place them at the right distance so that it looks like it's a certain color. Those colors blend in your eye, or it looks like it's a sharp image when it's actually a blurry one. So if you look at, actually there's so many things to look at, but I'd explore. Look at that paper, the way that paper looked. I have to explore a little bit. Take another paper, or here's a napkin. Love it. Just take a piece of napkin, paper towel. Remember how tight the fibers were on the paper? Totally different. This one looks like my hair when I get up in the morning. It's a mess. There's one fiber. Nice. Kind of translucent. See through it. I can go back to scanning. See what it shows me. Oh yeah. Awesome. Go a little bit more in. Why do you think it would be like that? Why would this paper have so many gaps in between? So a paper towel compared to a regular piece of writing paper. I would guess it's because turning on top lighting. What's the purpose of paper towel? That's a good picture. To absorb. So if you have some fun, get creative. Let's see how fast it absorbs something. I'm going to take the tip of my marker. I'm going to go in here. There's my marker tip. I can see it. It's not in focus yet. I'm bringing it down, and touch. Oh, you see the tip? Right there. Sure, my tip's getting in the way. I have to go at a better angle. Can you see the ink creeping? You see it moving, the ink bleeding. It's absorbing. You can see it over on that side. So you can see that the paper towel is actually sucking the ink into all of those gaps to pull it in. You know, I can see lots of wet ink just hanging out, filling in those gaps. It's a quicker picker upper. If I do the same thing with regular paper, the paper I just had. Let's take this back out. Pop it back under the scope. Make sure I can see, look at that, my colors. It's the surface of the paper. Here's the tip of my marker, which is kind of cool in its own right. Oh. Pretty sweet tip of the marker. Now I'm going to tilt down on the paper. See if we can get focus on that paper. It's like not even spreading. Why? Why would it do that? Why would it spread so fast on the paper towel and not so fast here? She's barely even leaving a mark. There's a mark. Focus. Super slow. My guess would be because the paper is so tightly packed, the ink sits on the surface and it doesn't absorb the way that it did with the paper towel. You see the paper. Try it with different things. Explore, get creative. Putting that page back so we can move on and get to other cool things. We got money to look at under here. Bugs to look at. Next, we're going to look at the letter E. So this just talks about pointillism. And there's the Marilyn Monroe, Albert Einstein image you can look at. Next page. Boop. Etch a sketch. So did you notice there's a kind of a weird pattern that happens when you're looking around the stage? When you try and push to the left or right or up or down, does it always go where you want it to go? Doesn't for me. So in your little envelope, grab black and white newspaper, which is another fun adventure. I've got the corner ripped off of mine. Let's see what that looks like as I slide it in. I'm going to look for the letter E. And that paper is pretty cool too. Love it. So if you really want to get a good image, I have my finger on it and it jumps a little bit every time that my pulse happens. So if I wanted to get a really good image, of course, I'd have to take these clips and put them back over and let go of the paper. So let go of it. Oh, and it fell. I'd have to make sure it was clipped on. But you're going to look for the letter E. Lots of letters in the text here. With my naked eye, I'm seeing the word 0. It says, and approved with 0 Democrat votes. Nice. There's my letter E. Okay, with this one, I'm definitely going to clip the paper down. Clip. Because I want to take the opportunity to draw the letter E. Because I want to understand what's going on when I move my scope around. Oh, I got pretty lucky that's not moved much at all. So is this the letter E you're familiar with? It's pretty cool seeing the print on the paper. Okay, so what I would do, I'm curious about how this E moves. First of all, if I look at the letter E, my naked eye, it was the word 0, right? It's like that. So when I'm looking at my E, draw my circle. This is my view, my field of view, FOV. And that E pretty much fills it. Oops. I've got a little of something else over here. It's kind of not perfect. So I'll take the time to draw. That's my letter E. What I want to do is make a note of what objective I'm on. I'm on the 4. So my objective is 4x. But I also looked up here to the ocular eyepiece at 10x. Remember? So I want to write both of those down. That's what my magnifications are. I'm looking at the letter E under 4 times and 10 times. So quick math, 4 times 10, my total magnification is 40 times. Pretty easy math. I'm going to leave the newspaper alone, and I want to look at it on the next power. So I'll take the nose piece, click, fine knob only. Oop. Holy moly, that was a big jump. So it looks like I'm only seeing the bottom of this bar. So my next one I'm looking at, usually it makes more sense to go in order down the page, but my next one I only see that line across here. And the only straight line on the E is the bar that goes across the middle of the E, so that must be what I'm looking at. Yeah. So it jumped from this area right here, somewhere, and closer. This is where it's going to get really tough. So I drew my little doodle. I do see there's a little more fiber here. So I'll just draw those fibers, just a little scratchiness. It's not about getting details of the drawing. It's about making notes of what you see. And for me, I'm seeing the fibers. I know that's important because it's something I wouldn't normally see with my naked eye. I just slid that bar down. Sweet. So I'm at, my objective is, it says 10 on the side, 10x, part I look through. is 10x, my ocular or eyepiece. So I'm looking at 100 times magnification right now. And I've got a better view actually. I can see some of those fibers that kind of go over to the side. Some long ones go with the black line. I got a little swooshy one there on the top. These are kind of just jumbled up. But now I can see that perfect letter E is not really as perfect as I thought it was. 100x. But I'm centered. And you can always come back and do this again with any other letter. Pause me. I'm going to turn my nose piece one more time, see what happens. There it is. Did you see it? Right there. Holy moly. Crazy. I'm guessing that swoosh that I noticed earlier is that. So I noticed the swoosh right through here. It's probably the same thing. A little bit fuzzy up here, a little fuzzy down there, and kind of shade it in. So this one, I'm looking at 40x for my objective and 10x for my eyepiece, 400 times magnification. So I looked at it at 40, 100, and 400. Oops, got a 0 magnification. And you can see a lot of human cells at 400 magnification too. So a couple of things you learned. Look at how things are getting bigger fast. You didn't actually make the paper bigger. All you did is made the image bigger. Another thing you probably noticed is when you slid the letter E to the right, which direction did it go? To the left. So moving right equals left image, or the image moves left, image moves left. But what about up and down? Did you notice? So when I went up, did it go down? Well, I can always go back to my shortest objective and check that out. So letter E again, bring it back into focus. Oops. Cool. So I'm going to grab paper and pull the paper down. Oh, it went down. Let's try this again. If I take the paper, move to the left, the image moves to the right. If I move the paper to the right, the image moves left. So kind of like an Etch-a-Sketch, left and right go backwards, up and down go where you want it to go. So you just have to retrain your brain a little bit. But it's fun just putting things underneath there, whether it's your fingertip, your sibling's fingertip. If you get a paper, or if you get a cut, put a drop of blood on there and check it out. I'll show you how to do that in another set of slides, or a set of videos. But whatever you're curious about, if there's something in the kit you want to look at it, play with it. The next page is text analysis. We actually started by looking at paper text. How about if we look at something that has way better print on it, like the bookmark that came with. So this is professionally printed, really nice paper. It's not just typical printer paper. So we can see the letters here. Let's see what happens. Let's go in over to the side where, what color do I want? I'll go here in the green. My scanning objective, red is there. Let's bring the paper in. Boom. Cool. There's the edge of the paper. There's the green just over the edge. Clear it up. Oh, it wasn't so green. Looks like a lot of yellow and some blue, some red. That's the green. Slide to a deeper green and see what happens. Looks like the blue and the yellows overlap more, giving me that green illusion. Let's go and see Rube's head. So I'm going deeper to the center of the bookmark. pretty far. Cool, there's a little yellow swish behind Rube's head. You see that? There's the side of Rube's head. Well, his head's red, and actually this is pretty red too. Nice. Look at the black coloring on his face. It's not all black, it's multicolored. We can zoom in on some of those. Oop. So I still haven't gone off of the top lighting. Lost it. That's no good. I'm going to go back to the last objective. And that's another thing to learn too, is that you may not use all 3 objectives to focus on something. It may be perfectly clear just using one. So I'm going to flip the light to underneath. Let's see what happens. It almost seemed like this color's moved. They didn't. There we go. It's pretty cool. So that's under lighting. There's top lighting. Cool. I didn't find the text. Let's see if I can find some text way up here. There's text. Looks like that's part of the word app. Zoom out a little bit. Is that an A or an E? Oh, it's E. It's the end of the word the app. Sweet. If you have pictures like family pictures on paper, I know most people have it on their cell phone anymore, but just try it. Look at pictures, photographs that were developed somewhere. See if they have a different method. If you get negatives, if you print photos, if your parents have negatives sitting around, try one of those under the microscope. That's the thing that's kind of like a brownish color, but it actually has a reverse image of the picture on it. Okay, so let's do some science experiments underneath here. Again, you don't have to just stick with looking at flat things. You can experiment. So the next page actually has one of my pages. There you go. Chemical chemistry. So you're going to create a slide and you're going to do a chemical reaction underneath. I need scissors for me, please. For this one, you will actually need some scissors. If you don't have scissors handy, you can always use a plastic slide out of this box. But when I'm doing something I think might tear something up, I like to use transparency paper without breaking anything. Thank you. Or you don't need to get scissors because I have another idea for you. That note card we used earlier, this one, let's just flip it around. So we'll use the other end of it, punch a hole in it just like you did before, just like that. But with this one, I want to make sure that the edges are pulled back. Okay, I'm going to punch another hole here. Same thing, and then the last hole over here. So now, I heard someone call this a business card slide because you can take someone's business card and if you're never going to look at it again, you just punch holes in it and use it for a microscope slide. So let me make sure that hole's fixed. Yeah. Flat, flat, flat. All right, take the tape like we did before. The only thing I don't like about Christmas. I like to wrap, but I don't like taking tape off, even when it's in that container with the cutter on it. All right, I'm pulling back enough so that I have multiples. And I'm going to pretend you don't have scissors, so I'm not going to use my scissors. All right, I'm going to cut it, snip, cut this into more pieces. 1, 2. So I just put a hole here and a hole down here so they can rip it in 3 parts. Well, easier said than done. There it goes. And here's the rough edge. And do like I did last time. Flip this over and cover the smooth side so nothing falls through. I have to poke this one more time. Yep. Or, had I been thinking, I could have just ran it over all 3 holes instead of ripping it apart. Just like I'm about to do now. Boom. That was smart. I have a well in the middle, one on each side. And I'm going to take 3 rings. So I want to make sure that that well is deep enough. I'm going to put some chemicals in here, and I don't want the chemicals to escape. So I have 1. I'm bending that paper back. 2. 3. All 3 wells. So now I can trap whatever's going down in that. And this lab says, oops, where my tab said, please don't close that window next time. There we go. Maybe a little faster. There it goes. Okay. So it says, chemistry is the study of matter, its properties, and how it reacts with the environment. This lab's about crystals, so we're going to look at something crystally apparently. And it says crystals are made of molecules or atoms that stack neatly like building blocks to form special shapes with special properties, kind of like if you were to stack dice. So some crystals come kind of square, some come in different random shapes. It doesn't have to be a perfect cube, it can be triangular or it can be long. It can have little edges poking out all over the place. Right? So dehydration is taking the water out of something. If we start with a wet chemical, which we're going to do just the opposite, we can remove the water and the crystal should solidify and turn into a crystalline material. So if I have something that would normally be a crystal and I had water in it, I can dehydrate it. Like if I had salt water, I could leave it in a dish and let it dry out, and on the bottom I would just have plain salt, probably by the end of the week, if it was thin enough. Crystallization is the process of removing the water and letting it turn back into the crystal it wants to be. 2 things we're going to look at. One's called an ionic compound, which means ions are anything that has a charge. So when you see symbols for chemistry and it says something like Na plus, just like a battery has a charge, or Cl minus has a charge. In fact, you put those 2 together, NaCl is table salt. So anytime you see that, those are called ions. They like to stick to each other when water's not around, but if water's around, they split. They scatter real fast. So if I take a little bit of salt and I put some water on it, it should just dissolve really fast because it splits apart. The other thing we'll look at is called a polar covalent compound, which ionic means it has a strong charge. These polars means it has a little bit of a charge, like the North Pole and South Pole, but not nearly as much. So if I put water on this and it splits really fast, what do you think is going to happen here? Probably not split really fast. In fact, it might need a little bit of help. So what we're going to do is we're going to see if salt and sugar, if we can tell the difference underneath the microscope. So let's pretend that you spilled. I did this actually several years ago. I was at a cooking dinner for St. Patrick's Day, and everybody there was making their own dish. And there was a big tub of salt, and there's also a big tub of sugar on the table. And it wasn't me, thankfully. I would have been really embarrassed, but the person that was cooking, she was making Irish soda bread, and she put her cup full of sugar in it when she was making it. And when it was done and we all tried it, we all wanted to kind of almost vomit because it was so salty. She took a big scoop of the salt, so a whole cup of salt in her bread, and it was not good at all. But let's say that you spill salt and sugar. How do you know? How do you know which is which? I gave you kind of a clue, and that's kind of how I set the rest of these labs up. I give you a little fun idea, something science-based, and then you can figure out what it means or what you want to do with it. And of course, I'll walk you through each of them. But based on what we just talked about here, if you had the salt and the sugar, what's one way that you can figure out which is which? Well, when we're talking about dehydration and rehydrating, we're looking at water. If I put a little bit of water on each of them, which one would dissolve faster? Should be the salt, right? So one way to tell would be the salt. Another thing that you can do is when you're looking at ionic, the plus and the negative, the strong ones, they stack like blocks, just like this. You have this water solution, this mixture of salt water. You take the water out and it stacks like a building block. Polar covalents stack a little bit, but not very well at all. They break apart really easy. It's like taking all these dice and trying to get them to stay into a nice organized stack. They don't want to do it very well. So if we look under a microscope, we should see a couple things. If we add water, we should see one thing dissolve really fast, but just looking at the crystals, we should see there's a difference between sugar and salt. So your envelope should have sugar in it and salt. There it is. My tabletop's still pretty dirty, but there it is. Science is ugly. Get used to it. Sugar, salt. So I'm going to shake them down. So the sugar and salt are all the way at the end. I'm going to get my spatula. And I'm going to get a little messy. So I can either scoop out with the spatula, or I'm just going to try and do a controlled pour. I have my glasses on, so I hope the controlled pour is doing really well. One of the perks to having the tape on the bottom is that when the salt hits the tape, it should stay put. And you don't want to fill this well completely up. You just want to sprinkle a little on it. Whoop. So you can see right through it. Right. Tap it. And that's the beautiful thing about the well is that the well holds salt in it. Yeah, I still have quite a few salt crystals. Okay, next one. Put a little sugar in the opposite side. So I need to label these. Salt. S-A-L-T. I'm going to go all the way to the other side. The pink. I'm going to put my finger in here so I can see it. Cool. Oh, yeah, I can still see my finger through it, so I didn't go too crazy. I'll label that one sugar. And if you want to, you can put, there are other materials in there. You can put a little pepper. Yeah, what the heck? Let's put pepper in another one. Let's see if pepper looks like either of these other two. I'm totally missing it. Okay. I got a few pieces in. That's where the spatula comes into play. Just brush what I want in. Cool. Spread it out a little bit. Now it's time to investigate. Let's go to, here it is. Right. We have stage all the way down. I'm going to start with the sugar. My objective is on the red. Whoa, or scanning. I can see the light going right through, and this is even better because I know my light's in the right place. And I'm going to slowly bring it up. Maybe going too slow. Here we go. Cool, there they are. Sugar crystal. What do you think? Perfect cubes? Not so much. Actually, they look kind of like a hexagon with a point on the ends. Let's see if I can find any others. Is that the way all of them are going to be? Those look like fractured hexagons with a point. That's cool. Can you see the surface? Those are a couple of those are really cool. Pardon me, a little tickle in my throat. So if I were to draw this, which is part of the notebook idea, say I'm on low power. Ooh, let's check it out on higher power. Okay, that's pretty cool too. You can see it's like slicing right through the layers. It's actually really cool. It reminds me of an ice cube, kind of. I'm going back so I can draw what I saw. I see the one I want. Sweet. No pun intended. It's really hard to focus on that one. I get as close as I can. And then on my notebook, I would draw, what do you see? I'll flip to the screen with the notebook. So I'm at 4x and 10x. 4x down here, 4 times, 10 times up here at the ocular. So I'm looking at 40 times magnification. And I see kind of this cool, almost like a tube. On the inside, it looks like it had bubbles on it. It's not a real tube. But they're almost cylinder shaped. And I know not perfect cylinders because I can see a sharp edge here. See another sharp edge here. So I'm sure it's more like a hexagon, well, almost like looking at the pencil, like that. But it doesn't focus. 40x. Now I want to go over and check out, we'll stop at Pepper and see what Pepper's doing in the meantime. I have to turn my slide. Lift up my spring clip. There's some Pepper. Pepper's a challenge. Probably because it's thick and the light's not going through very well at all. So let's turn on the top lighting this way, and now we can look at it a little bit better. It's almost fuzzy, but you can see the cracked or broken edges around a little bit. Here's the edge of the paper. Whoops. What is that doing there? Do you see it? That thing that looks like a dice, a die. It doesn't look very sugary. Doesn't look at all like the sugar did. So I'm gonna draw my pepper. Maybe I can go in a little bit closer. Flip the rotating, sorry, I keep saying rotating, revolving nose piece to one more objective. Ooh, too close. Yep, just too close. Come back out. That little cube is driving me crazy. Right, so same thing, I'm at 40x. And this first one was sugar. This one's pepper. It's that weird cube that I'm obsessed with. And it's weird, it doesn't look very dark, like black pepper. It's all pepper black. This weird rock looking thing over here. So I'm curious, I have some more pepper in here. I'm just gonna dump a little bit of pepper on the table. My book, sure enough, I only see a couple black specks. The rest are white in color. So I'm pretty sure the black speck is going to be pepper. I'm labeling it. This might be white pepper, but the cube, don't know what it could be. It's not sugar. Definitely not sugar. So I'll take a stroll on over to the next. We already looked at our sugar. Oh, there's another cube. Yeah, look how perfect the cube is. It's like a pair of dice. Huh. Looks like a white pepper, piece of a white peppercorn. Cruising over, cruising over. My blue ring is around my salt. Booyah, I think is what the kids say, right? Cool. So that's salt. Just figured out what was in the other pepper container. So we have all these little cube salt crystals. Salt is pretty sweet, but not that way. Wow, I wonder if I can nudge these salt crystals to go in a row. It's a salt rave. Do, 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 do. It's a party. Pretty tough. I don't want to spend too much time doing it. But if we know how many salt crystals it takes to get across our field of view, we can actually measure the salt. So I'm trying to get kind of close. Ooh, over pushed. Well, I'm just going to say I'm at 4D times magnification. I have one up in that top corner, one right below it, one kind of crooked below that, one big one. It's about at least twice, almost 3 times as long as the others. Weird. What's it doing in there? Maybe it's not salt. One that's kind of to the side, but if that one were nudged, it would go in and fill up that blank. I'm sure of it. And it looks like another one down at the bottom. So if I say that middle one's 3 long, it would be 1, 2, 3, 4, 5, 6, 7, 8. So about 8 salt crystals. Is it 40 times magnification total? So now I want to test that other thing. I don't know where I put my dropper. So you'll need your eyedropper. The other thing was dissolving. So I'm going to get eyedropper, glass water, Blow out, bring in. Perfect. I only need 2 drops. Right. When I do this, I'm going to rest both elbows on the table. Gives me control because my feet are on the ground. Each elbow is on the table. That's 3 points of contact, so that's really good. I'm hovering a little drop of water over. Bloop. Look at it. They're breaking apart already. We'll see the big one in the middle, see it dissolving. It's almost like it's ice in a hot water bath. There it goes. So we could build a really cool experiment and time how long it takes to do this. Is that some kind of weird creature growing there? We unleashed it from the salt. Okay. I don't think I'm going to get anything with pepper. So I'm just going to go straight across. Get sugar under there. Oh yeah. This sugar's going down. Ooh, that's a pretty sugar crystal. It's like rock candy. Boom. I still have some water in my dropper. I'm going to lean on the table so I have control. Hover 1 drop over it. Coming in for the kill. Boop. I don't know. That looks like it's dissolving kind of fast too. It's taking longer, I think. Makes it to really experiment with that though. Just like ice cubes. I'll take a Sprite, Moneypenny. With ice cubes in it. Real slow. Actually, I think we kind of hit the stopping point. So it'd be interesting to look up why it would stop melting. Why would it melt at the beginning and then stop all of a sudden? Maybe you could Google that. At what point does sugar stop dissolving in water? That might give you the answer. Is it changing? Maybe a little bit. But we actually watched nonstop the salt go. So, salt, was it ionic or is it polar covalent? Its shape, it was perfect little cubes, right? Which is what the ionic is. Ions like to split apart fast in water. The salt definitely split apart fast in water, so it's totally an ionic compound. The polar covalent still has weird shapes. It's not perfectly cubed anywhere. It's taking forever to break down. So that's polar. It doesn't like to split apart in water as much as salt does. Do you ever put too much sugar in like a drink and all the sugar that sits on the bottom? The only way to get that broken up is to heat it up, get it boiling. All right, and then that's all for the 101 section. With the 102, 101 is kind of like playing with things, not doing so many slides, but just playing with things. 102 is working more with the slides, learning how to build a slide, how to stain a slide, how to maneuver with the slide a little bit better, looking at things in 3D, looking at things with the scanning upper light, looking at things from below. Let's take a peek at this from below too. There's a special filter that I wish I will show you anyway. There's a special filter that looks like a pair of glasses. Oh, I won't be able to. Sorry, I have the slide on it. Nuts. But this is a polarizing lens, so it only lets light move in one special way. Maybe if I take two of them. One thing I can count on is that sugar is not melting fast. So if I take one pair of the glasses and put it under the specimen, with the light coming up through it, I take the other pair of specimens. You'll just have to wait to do this because you only have one pair of glasses. And put over it, oops, polarizing light. I'll have to adjust the bottom one. Oops. Oh no, I moved it out of the crystal. Hold on, let me find it again. Paper. Oh no, I lost my good one. That's okay, these will have to work. So there are some sugar crystals. Just slide this one in like I was talking about. Hold it right here. So it polarizes the light, which means all the random light waves, which we'll talk about later, go in different directions. If I turn this, can you see the colors coming out? Things that are transparent refract light, but they usually don't refract light perfectly. So they actually are holding the ability to work like prisms. Let's put the color up. Ooh, that was a good one. I just missed it. There it was. And sugar is a really good example of that. Sweet. I think I'm saying sweet too many times. Mm-hmm. Cool. So you take a break. I'll stop this recording. You can watch it later if you want to add on, and then we'll start 102 in just a few minutes. Oops, I don't want to end.